is Aleia and I'm here at the Miyashiro Lab at Penn State to talk to you about quorum sensing and the Squid-Vibrio symbiosis. A lot of people think that because bacterial cells are single-celled, they don't work together at all. In fact, it's quite the opposite. Just like eukaryotic cells in a multicellular organism, bacterial cells communicate a lot. Sometimes we refer to quorum sensing as how bacteria talk to each other. Personally, I like to refer to it as how bacteria gossip. So the real question of quorum sensing is, how exactly do bacteria talk to each other? A basic definition of quorum sensing is the way that bacteria coordinate their behavior by communicating through small signaling molecules. So essentially, the goal of a single cell is to work collectively with others to exhibit a gene that it wouldn't alone. Derived from the legal term quorum, which is the minimum number of people present to make something valid. A quorum for bacteria is the number of cells present needed to activate a gene. Now, bacteria obviously don't have eyes, so they can't just count how many people or cells are present. Instead, they use small molecules called autoinducers as activators to measure cell density. If you break down the word autoinducer, it makes sense too. Auto means self, so the bacterium is self-inducing. This means when one cell comes around and starts releasing the same molecule, another cell can recognize that signal and adjust gene expression. So, like I said, quorum sensing pathways are how bacteria gossip. Just imagine a single cell of bacteria with one molecule of autoinducer, and a single high school student with a rumor. Alone, neither can do much damage. However, as the population grows of either cells or students, both the autoinducer signal and the rumor spread. However, in a cell population, a gene is activated such as virulence factors that influence infectivity, whereas in high school, teenagers are just mean to each other. So yes, bacteria do talk, just in a different language. Quorum sensing was first discovered by Professor J. Woodland Hastings of Harvard in the 60s. He was studying the bioluminescent marine bacterium Vibrio fischeri, and he found that when in high enough cell density, the bacteria start to luminesce, and they communicate using small signaling molecules. So the symbiosis starts with the Hawaiian bobtail squid Euprydis and the marine bacterium Vibrio fischeri. The Hawaiian bobtail squid is a nocturnal animal that hunts off the shallow shores of Hawaii, and it found a way to use Vibrio fischeri's light as a camouflage. The bioluminescence mimics the moonlight from above, countering any shadow that would form below the squid, allowing it to elude any predators that would be hunting it. But what's really cool about this system isn't the squid. It's actually what goes on inside the light organ here where Vibrio fischeri is housed. Vibrio fischeri is the only bacteria that can colonize this organ, making it a great basic model for other labs like mine to study quorum sensing. But quorum sensing is a lot closer to you than you think. A lot of pathogenic species of bacteria use quorum sensing to regulate virulence, and they attack you only when they reach a certain number, making quorum sensing a new topic of drug development as well as a fascinating and applicable realm of the human microbiome. Thank you.